Assalamu alaikum. So in this lecture what we will do is talk about regulation of enzymes. Now that we know the different types, we know what enzymes actually do, it's really important to regulate enzymes and how they function and when they function and where they function. So we're going to spend actually 3 lectures just talking about regulation. Now, of course, if you understand the concept of how enzymes are regulated, you would be able to understand how other proteins are regulated as well. So there are different mechanisms of regulation, and that's why the topic is a bit complex. I've divided these lectures into 3, and each one would include different topics. So first we will start talking about expression of isoenzymes, okay, and what they do. Then we'll talk about regulation of enzymatic activity via inhibitors. Then in the next lecture we're going to talk about conformational changes of enzymes and enzymatic reactions. We're going to talk about allosteric, we'll talk about modulators, different modulators, inhibitors, activators, and so on. We're going to talk about reversible covalent modification and irreversible covalent modification as well. Then in the third lecture, we're going to talk about regulation of enzyme amount, location, and compartmentalization and complexing of enzymes. And we'll also talk about non-specific regulation of enzymes. So let's start. We'll talk about isoenzymes. They're also known as isozymes, okay, for short. So basically these are enzymes, different enzymes. They can act on the same substrate and they produce the same product, okay? So, and they are highly similar to each other, except that they are produced from different genes. So you will have different genes and they can be located on different chromosomes, by the way. So you can have isoenzyme B, C, D, E, and not only that, but they can also be present in different tissues as well. So you can have enzyme C produced in stomach, you can have enzyme B produced in muscles, and you can, by the way, have 2 isoenzymes produced in the same tissue as well. Okay? Now, the thing is that they have different catalytic activities. Okay, so for example, this isoenzyme, for example, can convert this triangle substrate into a circle substrate more efficiently than the reverse reaction, that is circle to triangle. Now, isoenzyme B, the second one, would actually catalyze circle to triangle more efficiently than triangle to circle. You see, isoenzyme C can catalyze either reaction equally efficient. Okay? Now, they're also regulated differently as well. So you can have, for example, this substrate, the circle, activating the enzyme. Now you can also have, for the second enzyme, the triangle inhibiting the enzyme. But the circle doesn't have any effect on that enzyme. You see, this enzyme is not regulated by either one of them. Okay, so they are regulated differently. So 1, they act on the same substrate, they produce the same product. That's 1. 2, they are produced by different genes. Okay, 3, they have different tissue distribution. Okay, 4, they have different catalytic activities, and they can also be regulated differently. So let's talk about metabolism first, okay? And this is an overview of metabolism or breakdown of glucose. So glucose is broken down all the way to pyruvate in 10 different reactions. In a pathway that we call glycolysis or glycolysis, that is breakdown of sugar. Now, the net result of this conversion of glucose to pyruvate is basically production of some ATP molecules as well as production of NADH, the electron carrier. Now, pyruvate has two fates, okay? It's in the presence of oxygen, so that's aerobic metabolism, it's converted to acetyl coenzyme A, and then acetyl coenzyme A enters the citric acid cycle, which is also known as the Krebs cycle, producing a little more energy as well as more electron carriers, and these get into the electron transport chain and oxidative phosphorylation, producing a lot of ATP. Now, in case there is no oxygen, pyruvate is reduced into lactate. Okay, this is the anaerobic metabolism now. This reaction is catalyzed by an enzyme known as lactate dehydrogenase. And this is going to be our story for the next 5-10 minutes. Now, so why is it that you have this reduction of pyruvate to lactate? In order to produce more NAD. And this NAD can sustain the glycolytic pathway that requires the presence of NAD. Okay, so producing more NADH that can be used for this reaction right here, but at the same time producing some ATP to keep the system going. So let's focus on lactate dehydrogenase now. Lactate dehydrogenase, LDH, is not really a single enzyme. Okay, it is actually the 5 different enzymes. All of them are tetrameric. So they are composed of 4 subunits, as you can see in here. So that's how it looks like. Now these enzymes, these isoenzymes, can be made of a combination of 1 or 2 protein subunits or polypeptide subunits. And these subunits are H and M. Now H is a characteristic of heart tissue, so it's produced in high quantities in heart tissue, and M is produced in high quantities in skeletal muscle. That's why it's given this designation, and it's also produced in the liver. Now you can have different combinations of lactate dehydrogenase. So you can have all H, all M, or you can have 3 + 1, 2 + 2, or 1 + 3. So you have different combinations, okay? But collectively, they all catalyze the same reaction. That is, lactate is oxidized to pyruvate, or pyruvate is reduced back to lactate. So lactate dehydrogenase catalyzes the reaction and its reverse. So again, they are produced from 2 different genes, okay? And you can have different combinations. So you can have the all H, which is also known as LDH1, or you can have the all M enzyme, which is designated as lactate dehydrogenase 5. Now, or you can have different combinations, okay? Now, these isoenzymes, they differ in their primary structure. So even though they are highly homologous, meaning that they have similar amino acid sequence and nucleotide sequence, DNA sequence, there are subtle differences, and these differences make them different in their kinetic properties. Okay, so for example, lactate dehydrogenase 1, the all H enzyme, catalyzes the oxidation of lactate to pyruvate, preferably. Okay, yes, it can catalyze the reverse reaction, pyruvate to lactate, but it prefers to bind to lactate and convert it to pyruvate. Now, the all M isoenzyme LDH5 does the opposite. It preferably catalyzes the conversion of pyruvate to lactate. Okay. Now, of course, they have these 2 enzymes, or these 5 isoenzymes, they have different tissue distribution, as we will see in the next slide. They have different affinities to their substrates. Okay, and they are regulated differently, and they have different isoelectric points as well. So for example, the M subunit has a net charge of -6. Okay, so it is overall negatively charged, and it has higher affinity towards pyruvate, so it binds pyruvate with higher affinity. Okay, on the other hand, the H subunit has a net charge of +1. It has higher affinity towards lactate, so it preferably converts again lactate to pyruvate and NADH to NAD to NADH. Okay. In terms of their tissue distribution, LDH1, the all H isoenzyme, is found mainly in heart tissue, and the LDH5 is found mainly in skeletal muscle and liver as well. Now we can see that these different isoenzymes are present predominantly in RBC for LDH2, lungs for LDH3, and kidney for LDH4. Now, so the thing is, so why is it that we have these different isoenzymes? Because they are present in different tissues, and these tissues can function differently as well in different conditions. So muscles, for example, can function anaerobically. Okay, so it prefers to convert pyruvate to lactate. On the other hand, heart tissues cannot. They function mainly aerobically. Okay, and that is why they do not like to convert pyruvate to lactate. Rather, what they do is that they take lactate and convert it to pyruvate so that they can get pyruvate converted into acetyl coenzyme A that can get into the Krebs cycle or the citric acid cycle. So you get it? So the idea here is that muscles convert pyruvate to lactate so that they can anaerobically convert more glucose to pyruvate than to lactate, producing some energy that can sustain muscles and their function. Now, lactate is released from muscles. It goes into the blood and it reaches the heart. Now, in the heart muscle cells take lactate and they convert it to pyruvate, and then pyruvate gets into the citric acid cycle and producing a lot of energy aerobically, of course. Okay, so that's the idea behind these isoenzymes. Not only that, these enzymes have different catalytic properties in terms of their preference of which reaction to drive, they are also regulated differently, okay? And they have different affinities towards their substrates. So for example, the M4 lactate dehydrogenase isoenzyme has low Km for pyruvate, so it has high affinity for pyruvate. So it converts it to lactate. And something important in terms of regulation is that it is not inhibited by pyruvate. Okay, so the substrate does not inhibit the enzyme. So the reaction is always going on. It's always happening. Okay, so making sure that pyruvate is always funneled to anaerobic metabolism. Now, on the other hand, the all H lactate dehydrogenase has a low Km for lactate. So it preferably binds to lactate, converting it to pyruvate. And it has high Km for pyruvate as well. So it doesn't have, this high affinity for pyruvate. And something important is that it is inhibited by high levels of pyruvate. So if heart muscle produces a lot of pyruvate, the reaction stops. The lactate to pyruvate reaction stops. Okay, allowing for lactate to be present for other tissues. So muscle tissues are not selfish. Okay, so they keep some food for other tissues. Okay. So let's talk about two other isoenzymes. So the first reaction in the glycolytic pathway is catalyzed by enzymes known as hexokinase or glucokinase. What these do is that they convert glucose to glucose 6-phosphate. Okay, so hexokinase is an enzyme that catalyzes the phosphorylation of glucose. We talked about this enzyme before. And there's another, and this hexokinase can also convert fructose and mannose to the phosphorylated form of sugars. Now, glucokinase is more specific for glucose, and what it does is that it specifically phosphorylates glucose to glucose 6-phosphate. So why is this reaction important? Well, it is important because once you have glucose 6-phosphate, the cell can decide on what to do with this glucose 6-phosphate. If it needs energy, it takes it through glycolysis so that it gets converted to pyruvate, and then you have production of energy, whether it is aerobic or anaerobic metabolism. On the other hand, if tissues have, especially liver, okay, and to some extent muscles, if they have a lot of glucose 6-phosphate and there's luxury in having this, of course, it can be stored as a glycogen. Okay, so it just happens that these enzymes, hexokinase and glucokinase, is that they are also present in different tissues. So they have different tissue distribution. So glucokinase is a liver and pancreatic enzyme. On the other hand, hexokinase is an enzyme that is present in skeletal muscle and red blood cells. Remember that the purpose of liver glucose is to balance glucose level in the blood. Okay, so if there is a lot of glucose in the system, it gets stored as a glycogen. If tissues, peripheral tissues like skeletal muscle, for example, heart, brain, whatever, if they need glucose, then the purpose, the function of liver is to break down glycogen into glucose 6-phosphate, and it can send it to other tissues so that they can utilize it for the production of energy. Okay, now the purpose of glucose in red blood cells basically is to produce energy through glycolysis. That's the only way by which they can produce energy because they have no mitochondria. Okay, so in red blood cells, glucose is converted to pyruvate and then pyruvate to lactate, okay, producing enough energy for red blood cells to function properly. Now, so here's a biological significance of this reaction. That is the phosphorylation of glucose, and that is glucose can get into cells, okay, via transporters. Now, once glucose is inside cells, it can be phosphorylated by hexokinase or glucokinase. Once glucose is phosphorylated, it cannot be transported outside, so it gets trapped. Okay, now liver has a way of converting glucose 6-phosphate to glucose, and then glucose can be sent outside of liver cells, but this system does not exist in other tissues. Okay, so as a result, you would expect the reaction that results in the formation of glucose 6-phosphate to be very slow and not very efficient in liver, allowing for some glucose to be present, so as in the form of glucose not phosphorylated, so that it can be sent outside to peripheral tissues. On the other hand, since in red blood cells and in skeletal muscles, the only source of energy, well, for red blood cells, the only source of energy is the phosphorylation of glucose to glucose 6-phosphate and its conversion into pyruvate, the phosphorylation reaction of glucose has to be highly efficient so that glucose is trapped and it can be used for production of energy. Okay, get it? And it just happens that the function of hexokinase and glucokinase serves the purpose. So it just happens that the Vmax and Km values of these two enzymes are different. So if you look at this plot right here, notice that the Km of hexokinase towards glucose is very low, meaning that its affinity is really high. It's about 0.1 millimolar. On the other hand, so basically, right? Remember, Km. That's Km is the substrate concentration, whereby velocity of a reaction is 50% of Vmax. So that's Vmax, 50%. That go down. This is the value of Km for hexokinase. On the other hand, for glucokinase, Km is really high. It's about 10 millimolar. So it's 100 times more. 100 times higher than Km value for hexokinase. Okay, so again, that's Vmax. Okay, 50% right here, go down. So that's Vmax for glucokinase. And Vmax is higher for glucokinase. Okay, so why is that? There's something different as well. Is that hexokinase is inhibited by glucose 6-phosphate, but glucokinase is not. Okay. Another thing is that glucokinase is activated by insulin and is inhibited by glucagon. Okay. So why is that? Okay. Let's put it all together. So let's say that you have little glucose in the system. This glucose should not be stored. It should be distributed to peripheral tissues. And as a result, glucokinase in the liver is not really active. Okay, but in muscles, it is. Okay, it's really active in muscles. But then if there is a lot of glucose in the system, now the Vmax for hexokinase is not really high, so that it does not consume this glucose. Rather, it keeps it for other tissues and for the liver, where it can be converted to glucose 6-phosphate and it can be stored as a glycogen. Okay, so hexokinase spares some glucose for other tissues and for liver to store it as glycogen. Now, the thing is, if glucose 6-phosphate is highly present in muscle tissues, it means that there's a lot of sources of energy, that's glucose 6-phosphate, and hexokinase is inhibited, so that glucose is not trapped in muscle tissues, and it can be kept again for other tissues and liver as well. Something else is hormonal regulation. Okay, so glucokinase is activated by insulin. So when's insulin produced? That's the hormone insulin. When's insulin produced? When there is a lot of sugar, glucose in the system. It means that the system is happy. Okay, so glucokinase is activated so that it can phosphorylate glucose in liver cells so that it can be stored as glycogen. Okay, and it is inhibited by glucagon. What is glucagon? It's another hormone that is released when we are hungry. When we're hungry, there is no need to store glucose. Okay, it should be distributed to peripheral tissues. And as a result, this enzyme glucokinase is inhibited so that glucose is kept for other tissues. Now notice when we are fasting in Ramadan, or when we do not eat breakfast, or we spend a long time not eating, notice that it's not the proper time to store glucose. Rather, what you see here is that it's hexokinase that is active, or that is more active than glucokinase. But on the other hand, when we're happy, when we eat, now red blood cells and skeletal muscles use whatever they need of glucose, converting into glucose 6-phosphate by hexokinase, but they spare the extra glucose for liver to store it as a glycogen. Okay? So it makes sense, right? Lovely. So let's talk about regulation of enzymatic activity now. Okay, so there are different modes, different approaches by which enzymatic activities are regulated. Now, remember the glucokinase and hexokinase are good examples of, and lactate dehydrogenase. These are good examples of how enzymatic activities are regulated. But now we'll talk about the means of regulation. So let's start with inhibitors now. Now, inhibitors can be reversible or irreversible. In other words, reversible inhibitors are inhibitors that can bind to the enzyme, inhibit its activity, but they can be released from that enzyme. Now, irreversible inhibitors are those that bind covalently, very tightly to enzymes, stopping their action. Okay, now all physiological inhibitors are reversible. Chemicals, drugs, metals can be irreversible inhibitors. Okay, but physiological inhibitors are reversible. Now, Now these reversible inhibitors, by the way, they can bind to enzymes and they can be released, and there are different types. They can be competitive inhibitors, non-competitive inhibitors, or they can be uncompetitive inhibitors as well. Okay? So let's start with competitive reversible inhibitors. What these inhibitors do is that they bind to the active site of enzyme. So here you have a substrate binding to the enzyme, and here you have an inhibitor. Okay, so then the inhibitor mimics, it looks like the substrate. So it binds to the active site and it prevents the substrate from binding to the active site. So what it does is that it competes with the substrate in binding to the active site, and that's why they are known as competitive inhibitors. Now, so the thing is, do they affect the Km or the Vmax of enzymes? Well, if you think about it, so here you have an inhibitor, it binds to the substrate, or it binds to the inhibitor, right? Now the thing is, if you add the inhibitor binds reversibly to the enzyme, okay, so it can be released, and the substrate can replace the inhibitor, right? So the idea here is that if you look at the Michaelis-Menten plot, that's the enzyme without an inhibitor. Now, this is substrate concentration. That's the rate of reaction. So that's the rate of reaction, hyperbolic, right? Now, in the presence of inhibitor, okay, is a fixed concentration of inhibitor. What happens is that if you increase the concentration of substrate, now the substrate will start to win the competition at high concentrations. So eventually you will get into Vmax. So half of Vmax, right here, 50. Look at this point. Look at these 2 points. If you go down, so that's with the inhibitor, that's without the inhibitor. So what the inhibitor does is that it increases the Km, but it does not affect Vmax, because eventually you will get into Vmax at very high concentrations of substrate. But Km is affected. You need more substrate to fill 50% of the active sites in the presence of competitive inhibitors. So if you increase the concentration of the inhibitor, okay, what would happen is that eventually at very high concentrations of substrate, you would get into Vmax. So go down 100, 50%, go there, go down. So Km is higher, okay, indicating that, you know, the more inhibitor you add, the higher the Km, meaning that the affinity is even lower. You need more substrate to fill 50% of the active sites. So that's in case you look at the Michaelis-Menten plot. What if you look at the Line-Weaver-Burg plot? That is the straight line. Vmax is not affected. Okay, it's going to be like this. It's going to be at this point. But Km is what? It's lower. Remember, these numbers right here, you need to remove the negative sign and flip the number. So in the presence of a competitive inhibitor, let's say that in the absence of inhibitor, it's minus 10. Okay, and sorry, so let's say that in the absence of inhibitor, it's minus 10. So Km is minus, that is minus 1 over Km is minus 10. Okay, and let's say that in the presence of a competitive inhibitor, it is minus 1. Okay, so Km in the presence of a competitive inhibitor is 1. Km without inhibitor is going to be 0.1. So it's 10 times lower than in the presence of inhibitor, meaning that affinity is higher. Okay, so just plug in numbers and work them out. So Vmax would be the same, but Km would be higher. How about non-competitive inhibitors? Meaning that these do not really bind to the active site, rather they bind somewhere else. And they can, what they do is that they change the active site. They can, they do not block substrate binding to the active site, but the active site is not functioning anymore. Okay, so do these inhibitors affect Vmax or Km? Now notice here you have enzyme plus inhibitor in the presence of substrate, or let's say enzyme plus substrate, you can have the enzyme substrate complex. Okay, in the presence of inhibitor, you will have the formation of the enzyme inhibitor complex. Now if you add a substrate, it can still bind to the complex, so you can have an enzyme substrate inhibitor complex, but you do not have production of products, right, in the presence of the inhibitor. So the idea is, what is affected, Vmax or Km? Well, if you look at the Michaelis-Menten plot, you can see the hyperbolic normal shape of plot. In the presence of inhibitor, it looks something like this. Increase the inhibitor, it's going down. More inhibitor, it's going even down. So what's happening here is that Vmax is affected. It gets lower, it's reduced, but not Km. Why? Because the substrate can still bind to the active site. So if you look here, that's 100, look at 50, 50%, go there, go down. We're talking about this point right here, okay? Now in the presence of inhibitor, that's Vmax, it's 50% less than normal. So go here, go to 25, so we're talking about this point, go here, go down, same Km. Well, look at this Vmax, go here, go down 50%, go here, go down, same exact Km value, but Vmax is affected. It gets lower, as if the enzyme does not exist, because this substrate, this inhibitor will keep on binding to the enzyme and it doesn't get released. Nothing takes it off. But remember, it's reversible, okay? Now, if we look at the Lineweaver-Burke plot, again, Km is not affected. It's the same in the presence of inhibitor, but the Vmax is affected. So let's say in the absence of inhibitor, it's 1. In the presence of inhibitor, this point is 10. Flip it, it's 0.1, right? Because that's 1 over Vmax. Okay, so you need to flip it. So Vmax is lower in the presence of inhibitor. Now, the third type of inhibition is known as uncompetitive inhibition. And linguistically, uncompetitive and non-competitive are the same. But to differentiate between the two types of inhibitors, non-competitive and uncompetitive, the different words have been used. Now, the idea here behind this type of inhibition is that the inhibitor binds to the enzyme when the substrate is bound to the active site. Okay, so whenever you have this ES complex, now the inhibitor can bind to this complex, preventing catalysis from going on, from finishing. So the idea here now, or the question is, what is affected? Is it Vmax or Km? Well, definitely Vmax is affected because there's no catalysis. The reaction doesn't go forward. Well, how about Km? Well, remember that the inhibitor binds to the enzyme whenever the substrate is bound to the enzyme as well. So if you close your eyes, open your eyes, the substrate is bound to the enzyme. Close your eyes, open your eyes again, the substrate is still bound to the enzyme. So it is as if affinity increases because the substrate is always bound to the enzyme. So what happens to Km in this case? It goes down. Okay, so here we have the Michaelis-Menten plot. So in black you see the plot normally without any inhibitor, so it reaches Vmax, right? Now in the presence of the inhibitor, notice that Vmax is lowered. Okay, well, how about Km? Km is now lower, okay, meaning that the affinity is higher, but it's useless. Okay, it doesn't, because the reaction doesn't go forward. So let's look at the Lineweaver-Burke plot. Plug in the numbers. Okay, you would see that Vmax, this point right here, is higher than this point. Okay, Vmax, this point is lower than this point because you have to flip it again. Okay, so it's lower. But how about Km? Km is also lower as well. Okay, meaning that the affinity is higher. But again, it's useless. Now let's talk about irreversible inhibitors. Now these are not physiological, okay? Now these irreversible inhibitors have different mechanisms of inhibition. So some of them, they mimic the substrate, but they prevent the enzyme from catalyzing reactions and they kill the enzyme, okay? So what they do is that they bind very strongly to the enzyme. Now, what these do, or there are different types of inhibition. You have covalent or different types of inhibitors. You have covalent inhibitors, you have transition state analogs, and you have heavy metals. So what they do is that they decrease the concentration of the active enzyme. Okay, they kill the enzyme. So let's talk about some examples in here. You have covalent inhibitors, okay? So what they do is that they bind to the active site of the enzyme very tightly, covalently, and they kill the enzyme. They prevent the substrate from binding to the active site, okay? Now, an example is a molecule known as diisopropyl fluorophosphate, or DFP. Now this molecule right here is a killer. Okay, what it does is that it's the same chemical that are used in nerve gas, in sarin gas. That is, it's the what they call them, chemical weapons. Okay, the same molecule, the same chemical is also used as an insecticide. Um, Now, what this molecule does is that it inhibits an important enzyme known as acetylcholinesterase. Okay, so what this enzyme does is that it prevents it degrades a neurotransmitter known as acetylcholine. Okay, so you have a secretion of acetylcholine in our brains, you have stimulation of a nerve signal. Okay, and then this acetylcholinesterase removes acetylcholine, so you can have renewal of the electric signal. Okay, now what this molecule does is that it inhibits the enzyme. So acetylcholine stays for a long time in the brain, resulting in continuous stimulation of the brain, of brain cells, of nerve cells, and that leads to paralysis. Okay, so you can see a person hit by DFP being having very forceful spasms, and then paralysis, then death. Okay. Now it also happens that this molecule inhibits other enzymes like serine proteases, like chymotrypsin, for example, or trypsin. But is that the cause of death? No one knows, but we know that mainly it affects acetylcholinesterase. Okay. Aspirin is another example of a covalent inhibitor. Okay, meaning so aspirin is the chemical name of aspirin is acetyl salicylic acid. Now, if you remember the function of aspirin on lipids, it inhibits cyclooxygenase enzyme, preventing the production of prostaglandins, and you studied this in the lipid lecture. Okay. So what aspirin does is that it acetylates the serine residue in the active site of cyclooxygenase. Now you also have inhibitors known as transition state analogs or substrate analogs. Okay, these are known as suicide inhibitors. Okay, because what happens is that these bind to the enzyme. The enzyme thinks that this is a substrate that it can do a reaction with. It starts the reaction and then it gets stuck. So it puts, for example, a hydroxyl group in here, it removes a proton from there, and then it tries to add or to modify another group, but then it cannot find it. So it gets stuck. It cannot go back. So as if the inhibitor kills the enzyme, and it kills itself as well. So that's why these are called suicide inhibitors. Okay, now usually, since we're talking about transition state analogs, now usually these drugs, if they mimic the transition state analog, they are not stable. Okay, so manufacturing companies, they try to design transition state analogs that are stable. Now, a great example of a transition state analog inhibitor is methotrexate. Now, methotrexate is a very famous drug that treats cancer. It's used for treatment of cancer, and it's also used in treating other diseases such as rheumatoid arthritis and so on. Now, what happens here is that methotrexate is a structural analog of folate, the vitamin. Now, this folate is a substrate of an enzyme known as dihydrofolate reductase. Okay. Now, dihydrofolate reductase is an enzyme that is needed to regenerate the active form of the vitamin. And this vitamin can bind to an enzyme that synthesizes nucleotides. And these nucleotides can be used for DNA synthesis. And if DNA synthesis stops, then cells cannot proliferate, specifically highly dividing cells like immune cells that cause inflammation, okay, and like cancer cells. So if we prevent cells from proliferating, okay, then these cells eventually would die. Now, methotrexate binds to dihydrofolate reductase because it looks like the substrate. Okay, it looks like a folate. So it binds to dihydrofolate reductase, but it kills it. And it binds to the enzyme 1000-fold more tightly than the natural substrate. Okay, so this is the mechanism of action of methotrexate. Penicillin, the antibiotic, the famous antibiotic, is also a transition state analog. Okay, so it looks like the natural substrate. Okay, so you look at it, it looks like it. Now, what penicillin does is that it binds to an enzyme known as glycopeptidyl transpeptidase. Now, what this enzyme does is that it synthesizes the bacterial cell wall. Okay, so what happens here is that bacterial cell walls, or this enzyme, the transpeptidase, would bind to penicillin thinking that it is the substrate. It starts the reaction thinking again that it is the right substrate, but then it gets stuck. It cannot move forward. Okay, so the reaction stops and bacteria die as a result of that. Okay. Now, there's a serine in the active site of the enzyme. What it does is that it attacks this group right here, okay, leading to the reaction being irreversible. Okay, so it links these two together, but then it cannot move the reaction forward. Heavy metals are also inhibitory to many important enzymes in the body. Mercury, lead, aluminum, iron. They can also bind tightly to functional groups or active sites of enzymes. Okay. Now, some of them can bind non-specifically, especially at high doses. An example is mercury. Actually, if there is a mercury spill in laboratories, they can close the whole building, by the way, okay, because it's very, very toxic. Now, what mercury does is that it binds to sulfhydryl groups of enzymes, and these can be far away from the active site of enzymes, okay? And they can affect the structure of the active site, so they kill the enzyme. Now, what enzymes are affected by mercury are not known. Lead also can be toxic as well. It replaces the normal functional metals in enzymes such as calcium, iron, zinc, and so on, okay, by irreversible mechanisms. So it causes developmental and neurologic toxicity by replacing calcium in certain enzymes, and it can affect the central nervous system as well as other tissues.